Thank you for giving me an opportunity to present my work, Rapid Clinical BRAF V600 Mutation Testing using Idylla Platform. In this talk, we are going to talk about the evaluation of the performance of Idylla Platform for single gene BRAF codon V600 testing, optimization of BRAF mutation assay for different specimen types, and establishment of assay performance characteristics for its clinical implementation. Activating mutations in BRAF lead to constitutive activation of BRAF and hence RAF, MEK, and ERK signaling cascades, which leads to the promoting cell proliferations and hence cell growth. Among the different BRAF mutations, BRAF V600 mutations have diagnostic and therapeutic implications for a variety of solid tumors and some hematologic neoplasms. Therefore, rapid and accurate detection of BRAF testing is required for clinical testing for various solid and heme tumors. V600 mutations, these mutations are diagnostic marker of hairy cell leukemia, Langer cell histiocytosis, and Erdheim-Chester disease. Therefore, testing is indicated for patients that exhibit clinical or pathological features when these conditions are considered indifferential. For therapeutic implications, V600 testing is indicated in melanoma, anaplastic thyroid cancer, colorectal cancer, non-cell small lung cancer, and Erdheim-Chester disease. When treatment is considered in continuum of care of patients. BRAF V600E testing is conducted as a part of NGS-based testing most of the times. However, sometimes these testing can offer challenges due to sample processing and limited sample or longer turnaround time, which is around from 14 to 30 weeks, and hence can delay the treatment of the patient or the diagnosis of patients. Therefore, single gene testing methodology can offer results in significantly reduced, can overcome these challenges and can offer results in significantly reduced turnaround time, which is around 1 to 2 days. Idylla TM is a fully integrated and automated molecular diagnostic platform that combines the speed and ease of use with high sensitivity and high multiplexing capabilities. The aim of the study is to familiarize with the assay and then validate it for clinical use. For familiarization run, 14 BRAF mutation assay cartridges were run, these runs include commercial control cell lines, different specimen types, input genomic DNA, and establishments of quality metrics for the qualification of the sample. Validation includes 69 BRAF mutation assay cartridges that were run for establishment of assay performance characteristics and limit of detection using serially diluted commercial control and patient samples. Different types of specimens were used for the study, which includes 1 to 5 micron FFPE tissue sections or 10 micron FFPE tissue sections ranging in different sizes with tumor celerity greater than 20%. Besides that, peripheral blood and bone marrow samples, cytolite fixed FNA passes, and cytolite fixed FNA washes are also utilized for the study. Genomic DNA extracted from these FFPE sections, peripheral blood, or bone marrow was used. The input material which is tested range from 50 to 100 nanogram. The current studied cohort used commercially available cell line control DNA obtained from Horizon Discovery. These cell lines include 5 cell lines which are positive for V600 mutations. And the VF for the mutation range from 50 to 1%. There is one cell line HD123 which is positive for BRAF V600K, pos at 5%, and a negative cell line HD249. Besides that, 43 clinical specimens were utilized in the study, which includes 19 FFPE sections, genomic DNA extracted from FFPE, FNA, FNA washes, and peripheral blood and bone marrow. These specimens have differential diagnosis, which include adenocarcinoma, anaplastic thyroid carcinoma, glioma, hairy cell leukemia, Langerhans cell histiocytosis, melanoma, and there are certain samples which are normals. These samples include in total 21 positive and 22 negative samples. These samples were tested prior by NGS-based method. Idylla system utilizes a single-use single specimen disposable cartridges for the testing, and it has a slot to place the input material. The assay consists of 3 allele-specific PCR reactions for amplification of V600E or G or V600K, R, and M, or wild-type BRAF. Tissue processing and real-time reactions processed in the single cartridge, which involve process of a specimen liquefaction, cell lysis, extraction of nucleic acid, followed by real-time PCR, data detection and data analysis, and finally reporting. This whole process includes 2 minutes hands of time, and turnaround time for the reporting is around 1 to 2 days. Idylla Explore portal is used to visualize the amplification curves and obtain these CQ values. RNSP is used as a sample processing control or an amplification control included in each well and is used for the qualification of the run. Tumor cellularity for the tested specimens used in the study range from 20 to 90%, whereas BRAF testing by NGS showed varied allelic frequency in the tested specimens range from 16 to 45%. During the familiarization run, 5 cell lines are positive for V600E mutations with a BAF ranging from 50 to 1% were run. This run shows the BRAF mutation, this run shows BRAF V600 mutation was detected down to 1%. 100% concordance was observed with the orthogonal method in between the tested specimens for the familiarization run. A CQ value for wild-type BRAF ranged from 30.6 to 35.86. Given the range of CQ values obtained and sensitivity of method to confirm the lack of V600E mutations, CQ value of V6 wild-type BRAF 32 to 36, a limit of detection of 5% is established. Whereas for the CQ value of wild-type BRAF less than 32, LOD of 1% is established. Samples with delta CQ value of greater than 8 for BRAF V600E or G or greater than 12 for V600K, R, and M considered to be negative for mutation detection. For the validation runs, this is an example of BRAF positive and negative samples as obtained by Idylla Explorer. These graphs are obtained from Idylla Explorer. In the positive sample, you can see that in these 3 wells, this red graph is reflecting the amplification of SPC control, whereas this black one in the well A is defining the wild-type BRAF, and in the well B, V600E to D positive is a graph for positive mutation, reflecting positive mutation. This assay defines the results in qualitative way, it just reflects the results is positive or not positive. It's for mutation or not. On the basis of the validation run, 100% accuracy is observed with the prior results. Limit of detection study is conducted using a serial dilution study using cell line or a patient sample at different dilutions 25, 10, 5, and 1% or 0.1%, whereas in patient samples it's conducted at 10, 5, and 1%. Depending on the sensitivity on the basis of sensitivity study, analytical sensitivity of the assay is 1 to 5%, dependent on the wild type BRAF CQ. 1% when the CQ of wild type BRAF is less than 32, and 5% when the CQ of wild type BRAF is 32 to 36. Reproducibility study is conducted using 6 specimens, 3 positive and 3 negative were used. Each specimen was run 3 times. Our inter-run reproducibility study showed 100% concordance for the tested specimens. In conclusion, Idylla BRAF assay offers rapid and reliable BRAF testing in routine and challenging specimens with limited material. It Analytical sensitivity of the assay is around 1 to 5% depending on the BRAF wild type CQ. Idylla BRAF assay demonstrated 100% accuracy in tested positive and negative samples. This assay has certain limitations, such as qualitative BRAF V600 mutation assay does not provide distinction between BRAF V600E or G mutations or between V600K, R, and M mutations. A false negative finding cannot be ruled out for specimen types where tumor cellularity cannot be assessed, for example, peripheral blood and affinity washes. With this, I would like to thank clinical molecular pathology team and the department chair, Dr. Khan. Thank you, and I'm happy to answer any questions.